dive in here. Primordial sea, oceans of nephrology, just to take a pee. So, renal physiology. Our old friend Hippocrates spent actually quite a bit of time looking at urine and trying to sort of read the tea leaves, or the pea leaves as it were, by trying to understand what it means when you have lots of bubbles in your urine, or what about when your urine is cloudy, or what about when your urine is very dark versus very light, or God forbid, what happens when your urine is a little sweet to the taste versus bitter. Yes, indeed, Haley. They would take a little sip. And this is where diabetes mellitus, the description of diabetes mellitus, comes from. People would be have such high blood glucose levels that they would be passing glucose into their urine. And mellitus, mellifluous, melodious, meaning sweet. The urine was sweet. Diabetes, the elimination of fluid, that was sweet. Diabetes insipidus, on the other hand, a totally different disease. That urine, same clinical presentation of diuresis, except the urine didn't taste good to this guy. He was committed. He was committed to the practice. Again, let's point out. So this was the burgeoning field now of uroscopy, trying to determine something about the state of health of the individual by inspection of the urine. Our old friend Galen, I think I maybe had mentioned this previously. He was the first one to identify the kidney as the primary functional unit of renal physiology, not by any manner. He would cut open the back of apes and clamp their ureters, and then watch their kidneys swell up, and concluded that it was the kidney that produced the urine. Couldn't have been fun for those poor animals. But it was much later. I included this not because it's so important in the history, but it's kind of funny. This guy named Thomas Bryan in 1655 railed against this, at this point, very mature field of examining urine. So here is a uroscopist of the 1600s. You were sick, you didn't get to go to Main General, instead you went to this guy. You peed in a glass container for him. He looked at your urine and then divined that you needed to find the bezoar of an elephant or something and dissolve it in a tea and drink it or something. You know, like they had these kind of bizarre sort of alchemical, magical pseudoscience around interpreting urine. Well, this guy here, Dr. Thomas Bryan, he said, "Enough! Wherein are newly discovered the old fallacies, deceits, and juggling of the piss-pot science, used by all those, whether quacks and empirics or other methodological physicians, who pretend knowledge of diseases by the urine in giving judgment to the same." So he goes on and writes this long tract defaming the uroscopist, or calling them the piss prophet. That's pretty funny. So Richard Bright, much later, as urology becomes a more mature field, he demonstrated the connection between proteinuria, meaning a protein in your urine, and what we see here as edema, or swelling of the tissues. And so he connected protein in the urine, edema, and terminal kidney failure. All right, so there was something wrong with the kidneys. Perhaps this person has high blood pressure, and you are damaging the nephrons in the kidneys, thus throwing off a bunch of protein in the urine and not being able to offload all of the fluid, interstitial fluid that's in the body. So they swell up. This is a person in acute kidney failure. She is not long for this earth. And this was the beginning of nephrology, understanding the connection between the fluid compartments. We talked about the fluid compartments a little bit when I talked about blood, the intracellular fluid compartment, and the functioning of the kidneys. So because of this guy's work, glomerulonephritis, or inflammatory dysfunction of the nephron in the kidney, was known eponymously for this guy as Bright's disease for a long time. He was from Edinburgh as well, which I don't know if I said this before, but in the 19th century, Edinburgh was kind of the seat of medical knowledge in at least the Western world at the time. So, and he was there. And then let's jump forward another century to last century to this pretty cheerful guy named Joseph Murray. He's actually a pretty remarkable man. So, we have this glomerulonephritis, this kidney disease, right? Kidney failure. How do we deal with it? Well, kidney is actually a pretty easy organ to access, to cut out. In fact, it's quite easy for you to live on one kidney, so perhaps you are interested in donating a kidney to somebody who is lacking one. And they tried it in 1909, the first kidney transplant, and they used animal kidneys first. It was a pig. They used a pig kidney. They figured, why not? Pigs are kind of like humans. It looks the same under a microscope in 1909. Why shouldn't it work in a human? Let's just give it a shot. Take a fresh pig kidney, hack it up, and then whisk it into the ER, or the OR, 10 minutes later, totally still viable tissue, stitch it in, and within a few weeks, it is a black rotting mass, and the patient is dying. And this is, of course, because of MHC, major histocompatibility conflicts. The immune system rejects the tissue. So in 1954, this guy, Joseph Murray, in Boston, is the first to perform a successful kidney transplant on identical twins. And here they are, the Herrick twins. I don't know which is who, but one of them's got kidney disease and the other's got two healthy kidneys. And so they're going to take one out of one and put it into the other. And here they are inspecting the first dialysis machine. And so what is dialysis? Dialysis is like kind of an artificial kidney. You're going to pump the blood out. You're going to have a rough filtration of the water, trying to remove the toxins, etc., and then pump the blood back in. We're looking at the first kidney machine here. And this is the surgery that was first performed in 1954, pardon me. And here, there was no tissue rejection because the one twin recognized the kidney as being completely genetically identical to itself. They had exactly the same genome, no problems. Okay, and this opened the door for kidney transplant. Now we have kidney transplants that come from any reasonable donor, and we use immune suppressive therapy to allow that organ to flourish. All right, so there's a little of the history that I love talking about so much. What does the urinary system do? If I were to ask any of you, I'm certain all of you would be able to tell me that it functions in both elimination of fluid, obviously, but also the excretion of various waste products, mostly nitrogen-containing waste products from the body, right? So excretion and elimination. However, these two functions, although more widely appreciated, are in some ways subordinate to this function, which is the homeostatic regulation of plasma volume, solute concentration. We've talked about this in terms of the effects of calcitriol on calcium, right? But there's lots of other solutes that are going to be under control of other hormones and other mechanisms. I'll give you a taste of that today and Friday. The kidneys help to stabilize the pH in the blood via excretion or reabsorption of hydrogen ion or carbonate. And then the kidneys, along with the liver, are implicated, importantly, in detoxifying things that you eat that you probably shouldn't have. So the primary components of anatomy here, we have kidneys that produce the urine. Ureters transport urine to the urinary bladder, where it's stored and then eliminated through the urethra. All right, so we're going to need to go through a little bit of renal anatomy so that we can have the physiological discussion. Okay, here is a kidney, and here's the ureter descending from the kidney. This ureter is the confluence of these calyces, major and minor calyces. They're just the tubes leading that are going to lead the urine to the single outflow right here. And each of these calyces, a minor calx, calx singular, calyces plural. Each calx is attaching to a renal pyramid. And thinking about the flesh of the kidney, it's divided into two parts. The medulla, which is a generalized medical term that you've come across already, which means the center of some sort of, the core of some sort of organ. And then the cortex, which is the outer layer. Man, it's hot in here. Is that me? It's like warm. So this medulla is broken up into two basic regions. There are these renal pyramids, which are separated, and you can appreciate why they're called that, separated by these renal columns that you see here. The renal columns are going to be for the passage of blood vessels up into this boundary between the medulla and the cortex. I think that's all the gross anatomy that you need. Let's talk a little bit about the blood supply here. So the arterial blood, this is just sort of a list of all of the vessels that it goes through as sort of a roadmap for you when I talk about one or the other of them. The most important ones to what we're going to be talking about here is right in here. This is the most important stuff because that's the fine capillary structures that surround the nephron. But blood comes in via the renal artery branches between these segmental arteries and then goes up through these interlobar arteries to these arcuate arteries. And these arcuate arteries are the ones that are at this boundary between the medulla and the cortex. All right? And it will be branches off of these arcuate arteries, these interlobar arteries that you see here, which are then going to give rise to these afferent arterioles. And we'll see what those mean in a bit. So then we pass through what is the capillary bed of the kidney and return back to arcuate veins and make our way back to the heart through there. I'm not glossing over this. We're going to go through it in more detail in a better picture in a moment. Okay, yeah, here we go. So 20 to 25% of our cardiac output is being run through the kidneys. 20 to 25% of our cardiac output. And that represents over a liter of blood per minute. That's a fair amount of blood. Yeah.
So does that mean that not all of the blood is run through the
all of your blood, presumably with time, and that's going to be an exponential function, right? But with time, all of your blood is going to run through the kidneys. It just means that for every stroke of the heart that you have, about 20 to 25% of that volume of blood that's being ejected from the heart during that stroke is going to get jammed through the heart. So if you look at the stroke volume and you compare it to the perfusion of the kidney is about 1/4 to 1/5 of that of a stroke volume. So that's how they're calculating that. Yeah.
I guess I don't even know if this makes sense necessarily because going through such a complicated loop where things are different lengths. But how long does it take for, I guess, the same molecule of blood to get from being in the heart to going through systemic circulation then back to the heart?
Mm-hmm.
I guess that would vary depending on what path it's taking.
That's a great question. It's a biophysical question that's a little off topic, and we can theorize about that after class, though. All right? It's interesting to think about, but we're going to move past that. All right, so the functional unit, the primary functional structure in the kidney is called the nephron. And there's two types of nephrons that we're going to find. There's one called a cortical nephron, and there's these juxtamedullary nephrons. Juxtamedullary, juxtaposed, meaning right beside, medullary, medulla. All right? And then a cortical nephron, of course, because it primarily resides in the cortex. And the juxtamedullary nephron is a little bit deeper. The glomerulus itself is right at the boundary, and then it has this long loop of Henle. They are identical in terms of a gross comparison between them. They have all the same parts. The proportion between one and the other is a little bit different. Exceptionally longer loop of Henle, or a descending and ascending loop, a nephron loop, in the juxtamedullary nephrons. So then we have these cortical radiate arteries and veins. This is also, I should go back and change this. This word here, interlobular, is antiquated. It's antiquated. That's not actually the word that they use anymore. I should change that in that diagram. But the correct terminology now is cortical radiate, and it's a little bit more descriptive and a little bit less easy to confuse with an interlobar artery, which also exists. So these cortical radiate arteries are then going to put off afferent arterioles, which are going to go into the glomerulus, and then there's going to be efferent arterioles that come out of them. And we'll see what those look like in a moment. All right, so a closer look at some of the vasculature in these nephrons. 85% of the nephrons in a kidney are cortical, 15% are juxtamedullary. I believe there is, what's the number? There's over 1 million nephrons per kidney. You have over 1 million nephrons per kidney. Is that per kidney or in a person? 500,000 per kidney. I don't remember. Actually, it's not that important. But we see the afferent arteriole coming in to this structure from the cortical radiate artery. And this structure here is called the glomerulus. And then it goes through this pressure, and there's going to be, this capillary bed, there's going to be a filtration of the blood. All right, this is like a first gross filtration, and then all this other stuff here is going to affect that filtrate. And then the blood comes out via an efferent arteriole. And then we have this capillary bed that overlays these various tubes, which we will dissect in more detail in a bit, and help to resorb a lot of the baby that got tossed with the bathwater when we initially filtered the blood there, okay? So afferent, we know what an afferent nervous signal is. It's something that's coming up into the brain Efferent is something that's coming out of the brain. It's just like affect and effect, right? You're affected or you cause an effect. Afferent arteriole is coming into the glomerulus. Efferent is coming out. All right? You got that? A little bit of language. So in these juxtamedullary nephrons, what we are calling the peritubular capillaries in the cortical nephrons are called the vasa recta. Vasa meaning vasculature, recta meaning straight. It's these straight vessels of the loop of Henle. All right? Very important. We're going to talk about what they do in countercurrent multiplication in a bit, probably on Friday. All right, so here's an overview. This is a very generalized overview. So what I'm going to do is give you a big picture, and then I'm going to go to some details, and then I'm going to go back at the end of all this to a big picture with the details filled in. All right. So afferent arteriole, we're going to filter the blood here in the glomerulus. We'll see how that works in a bit, and then blood comes out. This filtrate passes in to what's called the proximal convoluted tubule. Proximal because it's near this renal corpuscle. Renal corpuscle is the entire structure. The glomerulus is the knot of capillaries that's inside of it. This tubular fluid in the proximal convoluted tubule is going to be responsible for initially resorbing much of the water that was lost and a lot of the solutes, the important solutes like glucose or whatever, different salts. Sodium is important for all, sodium resorption is important for all of these processes. And we pass into the loop of Henle, this descending loop and an ascending loop. This ascending loop, we're using active transport to actively pump sodium into the tissue of the medulla. And this concentration gradient, this osmotic gradient that we create, helps to passively drive the resorption of water here in the descending loop. Okay? So this ascending loop, we call it the thick loop, is water impermeable. It's water impermeable. And because of that, and because of the active transport of sodium, it's going to drive the absorption of water through this water permeable descending loop. It's called concurrent multiplication. We'll go through that more at another point. Then the DCT, the distal convoluted tubule, we have the variable resorption or secretion of solutes and water and excretion of toxins. All of this is under the control of various hormones. All right, they don't actually know; they have not identified any hormones that control these processes, whether there are or are not. But there's some hormones that we'll talk about. It's called the RAA axis that control this, both in the distal convoluted tubule, then into what is called the collecting duct. And this collecting duct collects the tubular fluid from several nephrons, continues to adjust the fluid level in them under the control of various hormones like aldosterone, angiotensin II, ADH, antidiuretic hormone. We'll talk about that. Eventually, the urine, what is now the urine, comes to the renal papilla and enters the calyx to travel to the bladder. Overview? Yeah? All right, let's look at the renal corpuscle. I want to get through glomerular filtration rate today. Am I going too fast? I don't want to. No? Good. All right. Here it is. What a cool picture. This is a scanning electron micrograph of a colorized micrograph of a renal corpuscle. They're quite small, only 250 microns in diameter, but dramatically visible even under a basic light microscope. So when or if you get to get into an anatomy lab or a histology lab, it's one of the more interesting things to stick under the scope. And you see here presumably afferent arteriole coming in green, and then the blue would be the efferent blood traveling out of the glomerulus. It's not this high surface area to volume cluster of capillaries. Now, I've talked about that before, right? Surface area to volume, n-dimensional and n-1 dimensional interactions that guide structures quite a bit. This high surface area to volume here is just so that we can get a lot of juice per squeeze. We're trying to pack a lot of filtration power into the small volume of the renal corpuscle. Here's a schematic diagram. So blood coming in through the afferent goes through this knot, and the fluid is going to pass through these filtration slits that come then into the capsular space of the renal corpuscle, and then the blood passes out via the efferent arteriole. This filtrate then passes out through into this tube called the proximal convoluted tubule. So when you look at this, I want you to notice that there are vascular and urinary poles to the renal corpuscle. So blood comes in here, filtrate comes out over there. I also want you to know in this diagram, it's not a mistake that the afferent arteriole is larger than the efferent arteriole. Why might that be? Why might you guess that is the case? Just think critically. Yeah, no. Yeah, absolutely. There's less fluid. The fluid volume has gone down, hasn't it? So to maintain a reasonable blood pressure, the diameter of the efferent arteriole is smaller. And in fact, we're going to be able to control the fluid pressure in this renal corpuscle by vasoconstricting or dilating the influx or efflux from the corpuscle. All right. So again, getting a little bit more zoomed in on what we're talking about here. So if we were to take a transect, first of all, the afferent arterioles come in, and we have this epithelium, capillary epithelium, or endothelium, pardon me, that is itself wrapped in this visceral epithelium, which is called a podocyte. Now, a podocyte, we talked about serous membranes. This is not a serous membrane, but there's an analogy to a serous membrane because it forms a sac, right? And that's kind of what's happening here. There is these capsular epithelia that form this capsule surrounding the glomerulus, which then blends in to the visceral epithelium that's on the viscera, the organ of the glomerulus. And these epithelial cells are called podocytes. P-O-D, referring to foot, because they have these feet on them that have little toes, the way the anatomists thought of them. We see all the striping pattern in here. Those are just projections. They're called pedicels. They're projections of the podocytes that interleave with one another, interdigitate, giving rise to a high, so we were talking about surface area to volume. This is high perimeter to surface area. There's a high perimeter of slits spaces between the junctions of these cells, which is through which our filtrate is going to pass. All right? So there's all these filtration slits in here between the podocytes, adjoining podocytes, which are covering the endothelium, the capillary endothelium. Two proximal epithelial capillaries, glomerular capillaries, are going to be joined by a special cell type called a mesangial cell. Mesangial cells are able, through their contraction, to control the diameter of the capillaries in the glomerulus. All right, and they are under paracrine control through a structure right here. So this structure is called the juxtaglomerular complex. It has both paracrine and endocrine function. Paracrine meaning it's in situ. It's going to affect in situ contraction of the mesangial cells. This is the auto-regulation we'll talk about in a bit of the kidney. And then there's also going to be endocrine function via the production of a hormone called renin. And I'm going to spend a significant amount of time talking about renin in the next lecture. Questions on that? All right, so this fluid is in here and gets filtered out through these filtration slits. Big plasma proteins that are in your blood, and of course red blood cells, none of that should be in a healthy nephron able to pass through these filtration slits in the glomerulus. If you have glomerulonephritis, for example, very high blood pressure that leads to problems here, this can break down and you get proteinuria, plasma proteins passing into the urine. It's a sign of incipient kidney failure. So this filtration pressure is about 10 torr, 10 millimeters of mercury. All right, so a pretty modest pressure actually. It works at a pretty low pressure. Here's the filtrate with the various small solutes in what we don't call this urine yet. This is filtrate here, and then it's going to become tubular fluid, which we will amend to become urine as it passes along the course in the nephron. Right? So it's passing through the pores in what are called fenestrated endothelium. So these capillary endothelium are fenestrated. Fenestra in French is window. So they have these little pores or windows in the endothelium. And then laying on top of that are these filtration slits between the pedicels of interdigitated podocytes. Okay? And here's a pretty picture from the capsular space. Not much more. So this is, I have just 2 minutes left here. I will talk about this one and then we'll leave it here. So what are the forces that drive this? Well, we have the blood hydrostatic pressure. All right, so the blood pressure as it's reaching the glomerulus is about 60 millimeters of mercury, pushing out of the capillaries into the capsular space. We have an osmotic pressure, a colloid osmotic pressure, also called oncotic pressure. You'll see that term somewhere. That refers, ONCOTIC, that refers to a colloid osmotic pressure. And this is because there are no colloids. There should be no colloids in that filtrate. They can't fit through the holes. That's the proteinuria that Bright had first identified. Right? So there is colloid osmotic pressure driving the fluid back in to the capillaries. There's also just the ambient capsular pressure, the pressure of the fluid. It's about 18 in the reverse direction. That gives us a net of about 10 millimeters of mercury in terms of the filtrate passing out through the urinary pole of the renal corpuscle. If you have too high of a blood pressure here, you're going to it leads you to nephrosclerosis, kidney disease, glomerulonephritis. You begin to rupture these capillaries, you scar the kidneys, and you can get atherosclerosis of these kidneys that is going to lead to renal failure. We're going to spend, I'm done now, we're going to spend the rest of class on Friday talking about how the kidneys deal with this tubular fluid and help manage blood pressure and different osmotic levels of different solutes in the blood. Questions?